Welcome to my world. Another day, another time for knowledge acquisition. Knowledge is power, health is wealth. Today's topic is very straightforward. Your role in Staph and E.Coli resistance, AMR. When you're talking of Staph and E.Coli, these are the primary organisms that first created resistance to the penicillin that was discovered by Alexander Fleming. That year was a very remarkable year in that everything uh, everything was like no more problem. It was like no more problem for us. We have overcome these germs, these microorganisms, and as such we are free to live as we like. But there is a caveat. You are not to abuse that penicillin, but what we do, we abuse it, and before long these things happen. So what is really our role? We are different professionals, we are different people, we are from different tribes, but we all have played a role in creating antimicrobial resistance. So it is that role that we played that I am making effort to really expose, so that when you now know your role, the role that everybody played, regardless of who you are or what you are, the role you played in creating this antimicrobial resistance pandemic, we need to now do a reverse, roll it back, and possibly be free of these resistant organisms that can really cause serious problems when you are talking of problems, ailments, diseases that refuse to heal. It is only this antimicrobial agent that can help us, and vaccines. But when we are creating resistance, then unknowingly, I must say unknowingly, then we need to advise ourselves. We need to open our eyes to now know what we are innocently doing and causing us a serious reversal of fortune. So sit down. If you like my presentation, please press the like button, subscribe, and allow more to come out from me. Introduction. Alexander Fleming discovered penicillin as a cure-all against microbe. He warned everybody on resistance to penicillin during his Nobel laureate speech and the interview. As early as 1945, Sir Alexander Fleming raised the alarm regarding antibiotic overuse when he warned that the public will demand the drug, and then we begin an era of abuses. That is like more of a focus. Yes, his focus became a reality. And when you look at this structure of penicillin and even a similar antibiotic called cephalosporin, this is an idea of the penicillin structure that was celebrated before bacteria produced beta-lactamase, an enzyme that broke the beta-lactam ring, thereby rendering penicillin and cephalosporin useless. So it was targeting the ring. You can see that area of ring. The upper one is pentose, 5 ring, while the cephalosporin is exos, 6 edge rings. So with that idea, when we say a bacterium is resistant, it means that the antimicrobial is no longer effective against it. The first resistance to penicillin was reported in 1940 by Abraham and Chen, that an isolate of E. coli, not even Staph, produced penicillinase to nullify the antibiotic. A Staphylococcus aureus strain was also reported in 1942, so close to the origin of the discovery of penicillin. To produce beta-lactamase against penicillin, we later started having methicillin-resistant Staphylococcus aureus. So let's talk more on this. The best way to understand resistance is these two plates on the left and on the right. The clear zone of inhibition shows on the left that all the microbes are susceptible. But after 10 years, the same organism and the same antibiotic, there is no more clear zone of inhibition, which means the microbes have all become resistant to the antimicrobial agent. So, well, there are still some clear zone where if you look clear. Research, studies and finding. With extensive research studies, it has now been confirmed that bacteria transfer resistance to bacterial species that are not even common to them, that they are not the same. It's like talking about hybrid, that are not related to it by staying in the same environment. It is done through plasmid, extrachromosomal gene that is found on the bacterial cell that have many resistant genes in them, and also through the jumping gene called transposon that is responsible for virtually all evolutionary trend. When you see some people looking a little bit different or something looking different from the other, that is the responsibility of the transposon gene that is called jumping gene. And so the resistant gene can be copied and pasted in susceptible bacteria. Now let's look at that common bacterium, Staphylococcus aureus. Everybody, almost anywhere people tend to know it, probably because the name sounds so funny or probably so attractive. Staphylococcus aureus is one of the ubiquitous species of Staph aureus. Staph genus. The best way to categorize them is either coagulase positive, which are Staph aureus, or coagulase negative Staph. Practically all Staph aureus isolates were methicillin susceptible until 1961, when Jevons found 3 methicillin-resistant Staph aureus strains among 5,440 clinical Staph aureus strains in England. 50 years later, over half of Staph aureus strains became methicillin resistant. The acquired resistant gene, MECA, by horizontal gene transfer mediated by a mobile genetic element, transposon, which I had earlier explained as responsible for most of the evolutionary trend. So the resistant gene would probably attach to the transposon and it move. One thing we must accept is that this resistance is not limited to bacteria alone. It could move, it cut across all microbes, all kind of microorganisms, whether you're calling it parasite, virus, or fungi. That's why we have antimicrobial resistance, which should be the concern of all. Because, for example, chloroquine resistance in malaria is due to a mechanism in the parasite that pumps the drug out of it. Even vaccine resistance could be as a change of in configuration of the surface hemagglutinin, when you are talking of either the influenza virus or even bacteria Haemophilus. In African trypanosomes, it is a complete replacement of the surface glycoprotein so that both the T and B cells can't recognize them anymore. So we must understand that antimicrobial resistance is not, we are almost everything is made from the soil, and all to soil we will all return. So we must be Yes, the reservoir. Our environment, like I earlier mentioned, is one with us. The reservoir for E.Coli is in human feces and animal feces. So by defecating in the environment and dislodging of these animal feeds containing these microbial agents in diluted form, the opportunity of creating resistance happen. And from there, the disease is spread through soil contamination. Even the manure you have could contain many of these E.Coli, contaminated water, and the rest. And we do the transfer through food handlers, even food handlers too. Zoonotic infection, where insect bites could even transfer. For example, the Anopheles mosquito, we know how that transfers malaria. And you can see the interconnectivity. It's not only the network of this GSM that is interconnected. Everything in life is. Yes, I spoke about everything in life being interconnected. So you can then understand your involvement. We are all involved because we are all interconnected. So the involvement shows clearly why One Health is important. Is a topic
that is addressed from when you look at these four diagrams, more or less. People live closely together so they can transfer all these resistance. Changes in climate and land use can also result in the releasing these aerosols and then transferring it. The mobility, where we move from one area to the other, we transfer all these resistance. Animals are more than just food. They are also defecating, breathing wastage, their wastage and water, even their carcasses. They are all used to spread this resistance. So you can then understand the word sanitations and other things that yeah, this picture shows how possible we spread contamination. We contaminate from one lab to the other. It's very clear. You can see from animals, from the cow, the birds, the bees, water supply, and so on and so forth. How the network is from the hospital to where you find most of these resistant bugs. You carry them home and the resistance continues to spread. The environment, farmland, so on and so forth. That's true. Yeah, we are now talking about One Health, Nigeria. Nigeria is one country that has understood this network very clearly. So some great minds in Nigeria brought all stakeholders together, whether you are a doctor, nurse, veterinarian, any category of worker, even the environment. One Health and register it with the Corporate Affairs Commission in Nigeria to help in actualizing this reduction in antimicrobial resistance. The corporate entity is called AMRE, and you can see how beautiful the certificate is. Certificate of Incorporation.
Now that we know the role we play in creating this gigantic antimicrobial resistance, AMR is called all over the world, what are we going to now do? As a physician, you need to do the right thing. As a medical laboratory scientist, you need to do the right thing. As a veterinarian, you need to do the right thing. As a nurse, as citizens of any nation, we need to do the right thing. Our disposers of things, we must be mindful. We don't have to abuse drugs because we find it very helpful and then we are now abusing it or using it as prophylaxis when they are not made for such. So let us all reverse in our whatever we are unconsciously doing to now create an environment that will not allow antimicrobial resistance to escalate further than what we have. When you look at those pictures of not having any drug to treat even ordinary open wounds and your open wounds become untreatable, you would know that there is really fire. There is fire on the mountain.